Hello, everyone. It's a pleasure to be here. We're going to shift gears a little bit and talk about a technology that enables other therapeutic active ingredients to be delivered differently. So why do we care? 35 billion lost every year to various factors. How about immune rejection? This meeting, I listened to a lot of lectures yesterday about delivering cell therapy to solid tumors. Well, I believe that we need to think about delivering it differently. Long time ago, maybe 25 years ago, when I was a young pre-med student in Temple ER department, we had an old product called naloxone delivering through an IV. And a lot of patients would seize and nurses would get stabbed. So I was part of a brilliant team that thought, why not deliver it through an inhalation? And that became the birth of inhaled naloxone and a lot of different things that we now inhale. Over the years, I've been fortunate to launch a lot of drugs and been fortunate to be privy to a lot of new drug delivery technologies, hot melt extrusion, soft gel, but somehow a lot of therapies we talk about today are still platinum therapies, Taxols delivered through IV. And we also continue to deliver our precious cell therapies through IV for solid tumors. So we see clinical trial failures and side effects. On the plus side, we also work with a lot of really great products that were developed a long time ago and could use a reformulation, but they don't know how. So why delivery? I came to Likarda because I've had several companies in cell therapy space, and honestly, my assets hit the wall when I couldn't reformulate it or deliver it. Now, for CDMOs, I would love to see a CDMO that partners with new companies in helping them reformulate and create better products that can create a new delivery to improve formulation, that improve patient outcomes. Not just follows the tech transfer box. For pharma, we're expanding rapidly, but why not do things differently? Why not consider a different technology that will directly deliver cells to tumors? We have a proprietary equipment. We can talk about how it easily implements, but most importantly, it's a whole system solution, but we do need to be open-minded because it is different. I worked with many different delivery systems for cell therapies, and fortunately, they included allogeneic, which triggered the rejection. There are carriers that lack control. We're going to talk about the system that's able to deliver time delivery. We're talking about sustained release delivery. And really, we need to try something different for solid tumors and cell therapy. It's about time. So this is our Core-Shell Spherification for Likarda. We have tried it so many times. The company's been around for 9 years, and Likarda delivery system does not require immunosuppression, and there is no rejection because we do not use allogeneic, and everything we use is a biocompatible material. We call it smart inactives because long ago we have an inactive ingredient called gelatin that encapsulated, known as soft gel. Now we're looking at encapsulating cells. And honestly, I brought in prop because each one of these beads, for those of you that sit close enough to me, have about anywhere from 1 million to 10 million cells, depending on the cell type. And this gets directly injected into tumors. So we protect cells. We work with biologics as well. We have controlled, sustained release. Some of our implants can last anywhere from months to a year. We avoid immune response, and active ingredients are unaltered. So unlike having to reconstruct your construct and go back to the phase 1, we do not touch your active ingredient. We preserve the integrity of the original intent. We have many different lines depending on the cell type, depending on what we hope to achieve. And honestly, this has started out with a vision for encapsulation of islet cells for the treatment of type 1 diabetes in dogs. The company is in Kansas City, the capital of animal health, And now this product for type 1 diabetes in dogs is progressing to commercialization. We had a very successful exit, and we're focusing only on human health. And the reason why it's important, because we believe that enabling your cell therapies, your biologics to perform better, create sustained release, is going to make a difference for patients and the outcomes. We have over 45 clients, and I'm going to show you some examples, and 70% of our clients are repeat clients. This is what the equipment looks like. So it's a proprietary encapsulation equipment designed for global scale. It's very easy to implement at the end of any manufacturing process. And we're not a CDMO. We work with any manufacturing organization of your choice, so as long as they place the equipment. It's designed to custom release these cells. And we are also able to tune in the persistence and the immune shielding. Let me tell you, this is what it looks like now, the alpha unit in our lab that was created from a Buki machine. And the vision is the controlled GMP model. Actually, we're the interesting point of inflection for Likarda. When I first started to take the company over, it was, like I said, it was a therapeutic asset for animal health. And now with 45 clients in, we actually have grown too fast. So we're actually looking for partners and collaborators and strategic partners help us to expand and grow. We started out last year looking for money to build this GMP equipment, and this year we had signed multiple partnerships, and we continue to sign and look for strategic partners. Because in order for us to take in our next 45 clients, we're going to need to be part of a much larger environment. So picture's worth 1,000 words. Let me tell you about these 4 scenarios, and we have more if you want to talk to us. We're around today and tomorrow. So case study number 1, very exciting. This is why I was excited about Likarda. Like I said, it only took 8 years to get to this point, but we've encapsulated islet cells, human islet cells, for the treatment of type 1 diabetes. And you can see after the first 2 implants, the insulin has stabilized. So this CSS hydrogel bead is immune protective. So the goal is to create sustained insulin release and improve glucose control. Now, this is an example of where we compared our CSS technology versus allogeneic. We all know that allogeneic is what usually people associate with when they talk about hydrogel delivery. And we have numerous data to show this is liver cells that are notorious, very difficult to manage. How we perform, as a matter of fact, that purple bar is the only one that you can see performing in vitro and in vivo. This is a case study of interleukin loaded. So when we're working with cells, we basically can recreate that beautiful environment of Petri dish. So we can impregnate cells with interleukins or whatever microenvironment that requires for cells to be healthy and stay in for 2 weeks or 3 weeks or whatever the release profile our clients desire. So this is the outcome where improved cytokine availability sustained T cell function. And actually, our client chose that blue line because that was a best approach. We'll fine-tune the final delivery formulation based on what the client wants to see. As you can see, on day 11, the client was really looking for cell viability to match the profile for that cell type. This is a very popular example through our orthopedic surgeons who use stem cell injections. And as you can imagine, a stem cell injection into the knee is a very popular treatment, but we know it's a weight-bearing joint. And as soon as the patient stands up on it, and 14% of the cells is what remaining. That's why you can see with no treatment, you can see bone on bone, a pretty painful joint. With a standard therapy, that's what we're talking about. That's just injecting stem cells alone. And when we encapsulate it in our viscocell technology, the stem cells actually stay in and allow for regeneration. This technology is used on horses, and we're in clinical trial to use this in humans. We've done a lot of work in logistics and cryopreservation. It became a very hot area because we are able to ship our technology on dry ice, and they're actually working on ability to ship it at -20, and now working on a new study on keeping it at room temperature. Just think about being able to ship your cells as is without liquid nitrogen. That is a huge issue for our area, and as we're talking about 36% increase the CAGR score in the next 5 years of cell therapies, we're going to be using a lot of liquid nitrogen trucks, and we're going to increase the cost just by logistics alone. So I'm not aware of anybody else who are able to provide this level of support by shipping cells or any fragile material on dry ice or at room temperature. We have 54 patents, and we continuously create new patents with our clients. This CSS patent comes across 20 countries, and we are meeting with a lot of global clients, although our focus up to date has really been primarily on US. Something new we've done, which is we've taken our 9 years of failures, because everybody talks about success, and we trained an AI system, a proprietary AI system that guides us to better understand which formulation is going to be more appropriate for clients. So in the old days, it would take us maybe 3 to 4 months to come up with the right formulation. Now it would take us 3 to 4 weeks. Now we're using our proprietary data of what worked and what didn't work with outside publications that are available to understand which precursors to use, which hydrogel is more appropriate for this technology. Also taking into consideration what our clients' end goals are. We have been very fortunate over the past 3 years to have a lot of partnerships. We have co-published with Takeda. We have partnership with Cold Chain Technologies. Edge is our partner for animal health. And actually, the study is coming out with Gates Foundation talking about our ability of improving logistics and shipping fragile formulations at room temperature. This is our brilliant team. I have one word: resilience. We have pivoted, transformed, and we've been able to adopt the changes in the marketplace to really assure that this technology is available to anyone who wants to look at their therapeutics differently. We have brilliant scientists from cell therapy biologists to biochemical engineers at the lab, and we have a very strong and lean commercial team. So how does it work? A client usually comes to us, and they come to us with an active ingredient. We formulate their desired formulation, whether it is encapsulation, or we work with leukopaks or blood banks, not using encapsulation, just using our technology for logistics or cryopreservation. If we do encapsulate, we provide 2 or 3 different formulations, as you saw from that graph, and the client gets to choose which one performs according to their desired outcomes. And obviously, after that, we have either ongoing studies for in vivo, or the client wants to conduct the in vivo studies, and then the formulation precursors is theirs, and equipment will go into their manufacturing facility of their choice. I want to thank you for your time, and we definitely have a lot more studies to present. But if there's any immediate questions, I'm happy to answer. Otherwise, please find me or my team during this day today or tomorrow. Thank you.
Thank you, Likarda.